Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is William Duick from Business Review, and I'll be your host. It's our pleasure to have Biopharmaspec with us today, who will be discussing effective biosimilar comparability studies. Today's guest speakers are Dr. Andrew J. Reason, CEO and MD of Biopharmaspec, Dr. Richard Houston, Technical Director for Structural Analysis, and Dr. Mark Milchip, Technical Director for Physicochemical Analysis at Biopharmaspec. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you do disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. We'll allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. If you click on the survey widget, there are a few questions in there for you to answer in order to provide some feedback, and you can do this during or immediately after the webinar. If you click on the green resource list widget, you'll be able to view or download several PDF files relevant to today's webinar. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But for now, please allow me to welcome Andrew.
Hello, everybody, and thank you for taking the time out today to listen to us. I have a brief outline for the topics that we're going to cover today. I'm going to do a brief introduction on Biopharmaspec. We're a relatively new company, so that's just to introduce who we are and our history. I'm also going to talk about product development of biosimilars and give an outline. My colleagues are then going to talk through some case studies and some analytics to present the core methods that we use for assessing biosimilars and showing comparability, a short summary at the end, and as William said, we'll have a Q&A session at the end of the presentation. So Biopharmaspec is just about 3 years old. We were founded by myself and Professor Howard Morris. Both of us have a long history in biopharmaceutical characterization and were principals in the MScan group of companies that many of you will know. And our senior management team and a number of the senior individuals, including Richard and Mark, who are going to talk later, all were part of that MScan history. But essentially what Biopharmaspec does is provide structural and physicochemical characterization services. We have a long history in methodology, both chromatography and mass spectrometry. And I'll talk briefly about the experience we also have with some of the hot topics in terms of biosimilars in development. Our pioneering history started with Howard Morris in the late 60s and early 70s. And Howard was really the person who invented many of the protein chemistry, mass spectrometry, and chromatographic techniques that we use these days for characterizing biopharmaceuticals. In the late 60s, early 70s, he developed methods for looking at glycopeptide structure, for sequencing peptides at the same time, and through the 80s, the methods that we use for disulfide bridge analysis. And as well as applications, he's also been instrumental in designing and pioneering some of the high-end mass spectrometers, the high-field magnet mass spectrometer in the early 70s, but more recently the QTOF geometry that really has revolutionized intact molecular weight analysis, peptide mapping, and sequencing in parallel. As I say, Biopharmaspec was founded in 2014, but all the principals have a long history in these methods, particularly through the MScan group of companies. The services that we provide can largely be defined in a regulatory sense by ICH topic Q6B. I'm not going to dwell on Q6B because my colleagues are going to talk about some of these aspects in more detail later, but suffice to say that our services really provide the data that's needed for the structural and physicochemical analysis of biosimilars and biopharmaceuticals. As I mentioned, as well as the instrumentation, the protein chemistry, the carbohydrate chemistry, and the methodologies, we have a very significant experience with biosimilar products. We've been involved as a team in many, many structural comparability studies, And some of the products that we've worked on recently and are working on at present are on this slide. As you will no doubt see, it's dominated by monoclonal antibodies or antibody-related products. And it will be no surprise perhaps to see that in the case studies later, that the data is dominated by what we've obtained from antibody-type products. So in the next few slides, I'm moving on to biosimilar product development. What we've done is picked out some of the key aspects that we see in biosimilar product development, particularly when you're talking about structural analysis and physicochemical testing. The first thing that you perhaps need to do is buy some innovator product or a reference medicinal product, RMP. So what do the guidelines say about that? These are European guidelines. It talks about realizing what the brand name, the pharmaceutical form, the dose, the strength, but also says you obviously have to buy multiple different batches of reference material to be able to build up a baseline for the structural assessment of the innovator product first off and for comparability later. So once you've got your reference medicinal product, initial studies often center around defining the primary amino acid sequence. You need to know what the primary amino acid sequence is if you're going to encode for your biosimilar and produce your biosimilar product with the correct sequence. There are lots of companies that will source innovative product. That's one of the services that Biopharmaspec provides in terms of sourcing reference product. And from there we move to the first of the poll questions that William mentioned. So if you are developing or have developed a biosimilar, did you perform the de novo primary sequencing of a batch of RMP before beginning biosimilar development? And there are three choices for the answer: yes, no, or we obtained the primary sequence of the innovator, although not through your own sequencing efforts. And it's important to remember, if you make a selection, you just need to hit submit after you've made that selection so that your choice registers. I mean, what we are interested really in this question about is when people are sequencing the biosimilar, it's always one of our recommendations that you do that early on. You define the sequence of the RMP and the innovator so you have a baseline. Obviously, you're then building that up with other structural information as you go forward. But you have that sequence defined. You know what the DNA, the gene sequence needs to look like when you start encoding for and translating your biosimilar product. And you're starting from a point of knowledge. So if you can make a choice on that, if you've had experience with development and sequencing, we'll move on to the result. So, I mean, there are sequences that are in various databases. There are sequences that have been defined. So, I guess we're not surprised by that answer. But what we would normally recommend is that the sequencing is done fairly early on, so you've established that baseline and can continue with the development by reducing any risk involved with a not absolutely categorically defined sequence. So the next important aspect we've picked out is you've got your sequence, you know what you're doing, you're trying to choose your cell clone. What we're basically saying and recommending to people is that the days where you might make a decision on the cell clone relating to what cell produces the most material or is most productive are gone, really. You can now use some of the more sophisticated analytical techniques to get very, very good quality analytical information about the products from quite a large number of cell clones. We've certainly been involved in studies looking at more than 200 different products. You can get good quality analytical data to make decisions about which cell clone to choose from the analytics rather than the lower level electrophoretic tests and chromatographic tests, and perhaps decisions made on the productivity of the cell rather than the quality of the product. The other thing that people often forget about when developing a biosimilar product is that obviously for the RMP or the innovator, you are purchasing material that isn't in the final drug product formulation. These formulations often include excipients that can interfere with structural and or physicochemical analysis. So it's important to do an assessment early on in the development, find out which analytical techniques may be interfered with by these excipients, whether they be surfactants like polysorbate or tween, monosaccharides or amino acids, and then work out a method to purify the active ingredient for further analysis and thus remove the interference. That purification method has to be qualified, and you also have to show that the recovery is at a high level and there has been no significant change to the product through that purification method. We do have a recently published paper under the widget section of the presentation that everybody can access. It's just the thought processes that we go through when we encounter a new formulation or a new biosimilar product in terms of how you might go about purifying it, but also the sort of analytical tests you might want to use to assess and qualify that purification method. So you've sequenced your innovator product, you've defined your purification strategy, you've produced the biosimilar batch, which through the initial physicochemical testing and maybe some functional testing is looking like it should. And one of the first things you need to start thinking about then is the sequence of my biosimilar product the same as the innovator? Richard is going to talk in more detail about that going forward, but it's important to realize that you do need to show that the primary sequence of your biosimilar product is the same as the innovator product. Quantity is also important. You need to be able to reliably quantify your biosimilar as the manufactured product from your own facility and compare that with the innovator and know how much of these materials are going into functional tests and certainly into clinical assessments later in the development. The extinction coefficient is the way that biopharmaceutical products are quantified. The gold standard in defining extinction coefficient is amino acid analysis, which provides the protein concentration, and then the optical density, normally through UV absorbance at 280 nanometers. And the Beer-Lambert law, which you can see in the slide here, is then used to calculate the extinction coefficient. And from that point onwards, all you need is a UV spectrophotometer to be able to define quantity. People often ask us if theoretically calculated extinction coefficients are acceptable, and our answer is that they're not, and we've been through a number of examples where companies haven't perhaps performed the actual determination and regulators have asked them to go back and do that. So it's something we often talk about and recommend is performed so you can get some reliable quantitation of the products. So once you've done that, and once you've got through that and you've got that baseline information, you're producing more batches of biosimilar as that manufacturing process develops. Usually you're also buying innovator material as it becomes accessible on the market. And then you need to start thinking about showing that the biosimilar product is comparable with the innovator or the reference medicinal product. The guidelines, which Richard will talk a bit more about coming up, direct you back to ICH topic Q6B, which I mentioned earlier. But there are a set of analyses that you need to perform, and we'll be talking about how we look at that in some more detail going forward. The important thing with the innovator side of the equation is to analyze enough batches so that you're getting a true reflection of the variance of that innovator product. And that variance, that breadth of that product in terms of its quality attributes or its structural features, are where you have to fit in the biosimilar to show that it is then comparable. You need to be thinking about different batches, different expiry dates, and hopefully if you buy a number of different products, you are then encompassing different manufacturing sites for the initial drug substance, which is then formulated to create the innovator final product. So the full set of structural and physicochemical data needs to be assessed to provide a balanced assessment of comparability. Obviously, the data takes a bit of interpreting. The breadth of it is significant, and experience is very, very important in assessing the broad data set. What it comes down to in a perhaps a simple sense is knowing what is acceptable variation, or in our terms, experimental variation from session to session or analysis to analysis, and what is outside that expected variation, i.e. significant and a significant change. The other thing that's important to realize is we're only talking today about half of the equation. Any structural assessment of a biopharmaceutical and a biosimilar is no different, has to have a balanced structural and functional assessment. And these two together are used to assess comparability. And there are lots of examples where you have fairly minor but significant structural changes that have no effect on the function and are therefore less significant. We now move to the second poll question, and it relates to the background that we've seen and talking to our contacts and clients. There seems to be more companies outsourcing these days, particularly recognizing what I summarized in the last few slides, the experience and the knowledge of the products and the methodologies. So the question is simply, do you see outsourcing playing a bigger role in the development of biosimilars? Or will companies choose to invest perhaps and internalize these analytical activities as much as possible as we go forward? So there are two choices. The first one is really you think that there will be more outsourcing, and second choice is there'll probably be more internalization. And again, if you make your choice and hit submit, we'll move on to the answers shortly. But the other thing I guess that we've seen is a move towards using more niche companies to outsource. Recognizing that the experience and the breadth and the experience with the techniques, the methodologies, and the different products, but also having some idea of how the regulator is seeing data. I do want to stress that Biopharmaspec is not a regulatory consultancy, but we do get involved with the development of many products and see things coming back from regulators, etc. So we tend to use all that experience in really assessing the products and see ourselves as an extension of our clients' internal capabilities and provide consultancy and services over and above the analytics to help support product development. So just moving on to the answers, it's pretty even, I guess. Slightly more people see the insourcing, and that's I guess that's not too much of a surprise. What we hear might have suggested more outsourcing, but it would be good perhaps if we could work that into the Q&A session in some way to talk about that a bit further later. So just moving on to a summary to bring in the product development section. It's important to know that structural characterization and physicochemical testing is very, very important, particularly in the research and development of preclinical phases. It helps sequence and characterize the RMP. It can define critical quality attributes that you can use to assess comparability and identify. It can help you get good data to choose the correct cell clone for your biosimilar development, i.e. the one that structurally is closest to the reference product. And also it can help you with initial assessment of biosimilar product, because as well as showing comparability, you have to show the regulator that you've got control of your own manufacturing process, i.e. batch to batch, there is comparability within the manufacturing of the biosimilar itself. As we get further through the preclinical phases, more batches of RMP and biosimilar are analyzed to build up a good overall assessment of comparability. But it shouldn't be forgotten that even in the clinical phases where GMP batches are being produced, characterization can also provide further supportive data to continue the comparability assessment, or certainly to look in more depth at things that might change if there's a change in the manufacturing process, or certainly additional sites for manufacturing coming online. So in terms of the biosimilars, we do have a number of guidelines that we can go to. This slide is in there just to highlight the fact that the EMA guidelines, which are on this slide, the FDA guidelines, and pretty much all the product-specific guidelines, all end up either directly or indirectly sending you back towards ICH topic Q6B in terms of the structural and physicochemical assessment that you need to do. And Q6B, which we saw earlier, has an appendix 6.1 which covers the structural and physicochemical requirements, and there are 6 in each of the sections of that appendix. We obviously don't have time today to go through them all, but we've picked out some of the core analytics that we think are very important in defining structural and physicochemical testing, and those are the ones shown in green on the slide that you can see at the moment. Because we're talking about antibodies, we're also going to talk about aggregation, an assessment of aggregation, which appears in section 6.2.2 of the appendices within Q6B. And we're going to do that because antibodies do show a propensity to aggregate, and that's therefore a very important assessment that we need to perform. So just to pick out those core analytics, these are now the things that we're going to talk about in the case studies as we go forward. And I'm going to pass you on now to Dr. Richard Easton, who is our Technical Director covering structural analysis of biosimilars and biopharmaceuticals.
Thank you very much, Andrew, and good morning or good afternoon to everyone. Okay, so I'm going to be talking about points 1 to 4 on the slide you can see, really concerning just the protein and the carbohydrate at the very much the primary level, and then my colleague Mark will be picking up points 5 to 7. So if we just start with the first point, the amino acid sequence, the text that you see on the screen there is taken directly from the ICH Q6B guide. And really what this is talking about is bringing together various different protein data sets to support the sequence that is shown through the DNA. This is very much a technique that has been used successfully for a number of years, but with procedures and equipment that's now available, there is much more capacity to carry out the full primary sequencing at the amino acid level of proteins and glycoproteins. As far as biosimilars go, if you look at the EMA guideline, as you see on the screen at the moment, this is the same one that Andrew showed earlier, Section 5.3.1 of the guideline actually states the target amino acid sequence of the biosimilar should be confirmed and is expected to be the same as for the reference medicinal product. So certainly the EMA is very aware of protein sequencing and the need for it. This statement is echoed in the FDA guidelines on their guide for industry as far as biosimilarity goes. Section 7 Part A states a very similar thing. It's expected that the expression construct for a proposed product will encode the same primary amino acid sequence as the reference product. So how do we actually go about achieving this? Well, there's 2 ways that we can do this, and I'll just flick on to this slide here to show you that. We can use gas-based sequencing, and we can use that in conjunction with or separate from mass spectrometric sequence analysis. The gas phase sequencing is very much a chemical method using the Edman degradation process, which is cyclic chemical removal of individual residues from the N-terminus of a protein or a peptide. The mass spectrometric approach is very different in the sense that that allows you to generate sequence data from fragment ions of peptides as they're generated. So in the next slide, this just describes a little bit of the process as it would take place. For us here at Biopharma Spec, we use Q-TOF geometry instruments, as Andrew alluded to earlier, in conjunction with LC separation. These type of instruments allow us to generate data that indicates or demonstrates peptide mass, but at the same time is also capable of generating data that produces fragment ions from those peptides as they're looped from the LC column. This is so-called MS to the E data, and that allows us to generate sequence data as we're actually producing peptide map data at the same time. And I'll talk about peptide maps briefly in a minute. We can also perform data-dependent acquisition, whereby we can specifically select peptides for MS/MS sequencing if those are required. The type of sequencing we do usually requires anywhere between 3 and 6 different protein digests. The reason for that is to produce a range of specific and well-defined peptides, but peptides that will overlap across the digests such that they can be linked together through overlapping regions of sequence. Now, mass spectrometry doesn't have all the answers as far as amino acid sequencing goes. There are amino acids that have the same masses, so-called isobaric amino acids, as far as the fragment ions go. Glutamine and lysine are 2 of these. Those can be distinguished readily from the way they react in different specific digests, such as the use of the enzyme trypsin, which cleaves specifically on the C-terminal side of lysine or arginine. Alternatively, isoleucine and leucine, which do produce the same fragment ions, are readily separated and identified through the use of the Edman chemistry technique. So it's possible to purify those particular peptides that contain leucine or isoleucine residues, and then these can be sequenced through Edman chemistry, and the identity of those 2 amino acids can then be found. So bringing those 2 different techniques, mass spectrometry and Edman chemistry, together allows us to obtain the full amino acid sequence of a protein of interest. So we come to another poll question now. If you are developing or have developed a biosimilar, did you perform the de novo primary sequencing of your biosimilar either at the beginning of the project, as soon as that was chosen which clone to use, or after some initial characterization had been performed, or later in the project during or after some comparability studies? And again, if you could just make your selection and press submit in about half a minute or so, we'll see what the results are. And really from this question, we're just interested in seeing what the thought processes are with regards to characterization. Obviously, working out the protein sequence, the full amino acid sequence of a protein or a glycoprotein, particularly something like a monoclonal antibody, is far from a trivial exercise. But having said that, as Andrew has mentioned earlier, it does give a very important baseline from which to work. It's fundamental to understanding your clone and knowing that you're starting off with the right product. So we're just interested to find out really what people's thought processes are as far as that goes. Okay, so we'll just have a quick look at the results of those. And, well, that's very good to see. 100% at the beginning of the project, which is very good. That's what we like to see. Yeah, that's nice. No one has chosen performing at a later date or later in the project at all. So, yeah, I don't think you could get much more clear cut than that. Okay, so moving on to the peptide map. Again, this is a text taken directly from the ICH Q6B guidelines. And a variety of different techniques are listed for identification of the peptides here. all following digestion using suitable, that is appropriate enzymes or chemicals for the protein of interest. We would use, or we do use, mass spectrometry as the most useful technique for peptide mapping. Again, in conjunction with LCMS, it does provide us with a lot of data that carries a great deal of weight, an advantage in terms of structural characterization. So that's really the go-to technique for us. And we'll talk a little bit about the data set as we go through. But first of all, how do we actually do this as a technique? Well, this schematic here will just show a little walkthrough of the methodology. So we start off with our protein of interest, in this case a monoclonal antibody. Now this may well need to be cleaned up first of all because excipients that are formulated with various samples can interfere. Andrew's mentioned tween earlier, and that is one that does cause problems with mass spectrometry. So a front-end cleanup may be needed. We then reduce the molecule to separate the chains, the light and heavy chains. We then carry out a specific protease digest to produce proteins of expected sizes. They may be glycoproteins as well. And then these are separated out by online LCMS, and the peptides that we find we can identify. The next slide just shows an example of some of the data that we obtain. The top chromatogram that you see here, labeled reference product, is just, as it says, a reference product, a representative chromatogram of a specific digest. Below it are stacked 3 biosimilar traces. An initial visual inspection will show that all 3 traces look very similar to the reference product. However, if you look at the doublet at 11.5 to 11.6 minutes, you can see that there's variation between the 3 batches and the reference product as far as that doublet goes. And indeed, biosimilar batch 1 is looking very comparable, but the other 2 batches are less so. So this is very useful as a visual aid to give you some idea of how similar your batches are as you're producing them or not. We'll come on to the reference product and biosimilar batch 1 shortly as well, with a little bit more information on those. So if we look a bit more detail about the data that we generate from the mass spectrometric analysis of these peptides, at the bottom of the 2 traces you see here, again we have a representative chromatogram of data generated. The upper trace is a computer search of that data for the signal at 660 Daltons, which represents the C-terminal peptide of this molecule. If we just look at the next slide here, the overlay on the chromatogram shows the spectra that are generated from under that peak that we found for 660 Daltons. The lower of the 2 traces is labelled the low energy mass spectrum, and this you can clearly see has the signal at 660 and very little else. The trace above it, labelled the high energy mass spectrum, is a so-called MS to the E data, which is generated at the same time by alternation of energies within the system as we generate the low energy data. This gives us fragment times that we can use to confirm the identity of that peptide and generate amino acid sequence as well. And the sequence that we do find matches for that C-terminal peptide, so we know that we have a positive identification of that. The peptide map is also useful in other ways because we're using mass spectrometry with its very high sensitivity for detecting any differences in mass. We're also able to look at modified forms of the molecule, which is another requirement of ICH-Q6B. They look for variations, post-translational modifications such as glycosylation and phosphorylation, and other protein modifications such as deamidation, disulfide bridge scrambling, and oxidation. Mass spectrometry is able to identify quite clearly deamidated peptides from the non-deamidated forms, even though the mass difference is only 1 Dalton. Okay, so we move on to another poll question now. Regarding the sequence data available from peptide mapping and the sequence data available from primary amino acid sequence, is it clear the difference between the 2 data sets and the information they provide, or is it not completely clear what the difference is between the 2? The beauty of using the QTOF mass spectrometer system is that we can generate the amino acid data at the same time as we generate the peptide map. It really comes down to the depth of data mining that we carry out on that. So it's a very useful system for actually generating both data types, and we can fill in any gaps that we haven't found either with other digests or with incorporating the gas phase Edmond into that. So we'll just take a couple of seconds more and see what people's thoughts are and see whether there is a clearly understood distinction between those two types. Okay, so that's exactly 50-50 between those two. So I guess we'll have to work to make that a little bit clearer for people as well going forward. Okay, in order to try and keep on time, we'll press on. So the next part I'm going to talk about is the carbohydrate profiling. Again, this is the information text provided in the ICH guidelines. It requires ultimately a full characterization of the glycans and the monosaccharides that are used to form them. There are a variety of chromatographic and mass spectrometric techniques for this. In the interest of time, we're not going to be talking about the mass spectrometric ones. We're going to focus on one technique, which is the 2-aminobenzamide fluorescent tagging of carbohydrates with LC separation and identification of those glycans. So how do we perform this? Well, we take our glycoprotein, and our antibody is shown again here. We reduce it and this is a traditional route, we reduce it to break the disulfide bonds. We carry out our specific protease digest again. This time we carry on and use the enzyme PNGase F, peptide N-glycosidase F, if we're looking at a mammalian glycoprotein, to release those N-glycans. The N-glycans are then separated from the peptides, and the glycans are derivatized by the 2-aminobenzamide label, and they're separated by Hillick chromatography with fluorescent detection and also subsequent MS analysis of those compounds. Nowadays, there is a more rapid way of doing this with buffers and various chemicals that are available, and it's possible to release those N-glycans directly using PNGase-F on the intact molecule, which certainly does save some time in the analytical process. The next slide shows some of the kind of data that we generate. This is an LC map. Some of the glycans, but not all of them, are clearly labeled, and this is from the reference product that you saw earlier. If we compare that data, and that's shown here, with the data from our biosimilar 1, you can see that this component here, labeled G0, is increased in the biosimilar compared to the reference material. That wasn't clearly shown in the peptide map, it wasn't readily detectable, but that could well have an influence on the ADCC activity of the biosimilar compared to the innovator, and therefore that would require some kind of bioassay assessment of that. Okay, the last part I'm going to talk about is the intact molecular weight. Again, for this we use mass spectrometry, which really is the gold standard as far as intact molecular weight analysis goes, because of its accuracy. Other techniques are available. We perform this in the following way. Take the sample, possibly clean it up if needs be, because things like, as we mentioned before, detergents can be a problem. If small amounts of excipients are present, then they may well naturally clean up on the LC column as we do the analysis. We can generate data on the intact mass, certainly for antibodies using QTOF instrumentation. That's quite an easy thing to do, even at 150 kilodalton mass range. We can reduce the molecule to light and heavy chains. We can carry out LCMS again on those separated chains and obtain mass for them as well to confirm that they are as predicted for the amino acid sequence. If we look at in slightly more detail at that, for an intact antibody we have masses around about 148 kilodaltons here. 3 main components are present. The mass differences between those 3 are consistent with a single hexose residue within the carbohydrate chains, which are showing the variation on the antibody. This is the kind of profile that we'd expect. The masses fit perfectly for the amino acid sequence along with the expected glycan chains. If we carry out deglycosylation of those molecules, we can collapse those signals down to a single mass which fits for the amino acid sequence of the light and heavy chains as predicted. Reduction of the intact antibody gives us heavy chain signals. Again, you see 3 components here, 2 major, 1 minor. The mass differences, again, are consistent with the amino acid sequence, varying only in the hexose residue, a single galactose residue on the glycan chains. Finally, the light chain data, a single signal fitting exactly for the predicted amino acid mass of that sequence. And at that point, I will hand over to my colleague, Dr. Mark Millichip.
Thank you, Richard. I'm going to carry on now to discuss the final few options that Andrew first spoke about with regard to the physicochemical analytics and some of the core methodologies that we would recommend. So first of those is the isoform analysis, again, reference to ICH Q6B. I've heard it said that this requirement from the ICH guidelines wins the prize for the shortest one because it's the clearest and just one simple sentence. However, with most of these things, the devil is in the detail. And although it mentions isoelectric focusing, it also mentions other appropriate techniques. And as Richard said before, when he was talking about the intact mass spec has taken over fantastically with regard to looking at intact mass compared to some of the other techniques. I have to say, for the other physicochemical properties and techniques, there are other techniques that have taken on or moved on since the ICH guidelines produced their recommendations, and these are some of the things we're going to be highlighting in the next few slides. With regard to the isoform analysis, this is talking about charge differences and variation in charge, and instead of using just isoelectric focusing, which is a gel-based assay that then you have to stain the gel, and there are various problems with regard to that. We're talking about quantitation, and you have to use a densitometer, and there are differences with staining and destaining, and it gets very complex. What we use is imaging capillary isoelectric focusing unit, a CIF unit, and I have to say, I think this has revolutionized what isoelectric focusing can do for you at the moment. Basically, what happens is it's isoelectric focusing in a capillary, but it has a UV-based quantitation method. So it's a relative quantitation method. So you can detect the relative amounts of any variations that you will see. And on the top here, on the left-hand side, what you see is the overall trace that you would see with the PI markers at the very left and the very right. And then in the middle there, you see your product. And if you then zoom in on your product and you then look at the trace on the right-hand side, what you see is your main product, your highest peak is on the left-hand side, and then you see these 2 smaller peaks on the right. And these are more basic variations. So you're seeing an increase in the base units, and so the PI has moved across. Now, further analytics have been performed on these samples, and like cation exchange, and it's been shown that this is a monoclonal antibody, and in this instance, these are the additional lysines on the C-terminus of the heavy chains. So this is 1 heavy chain and 2 heavy chains, and these are the lysines that are there. Now, this is quite indicative of you overdriving your cell production. Yeah, the cell's natural enzymes, the carboxypeptidase, cannot cope with cleaving off these additional lysines. So once again, this is quite a good indicative assay to show you how you're controlling and looking after the process that you're trying to develop with the cell line that you've got. Now, the next set of slides, again on the left-hand side, you can see the PI markers left and right. Again, this is a different sample, and what we're seeing here, more to the main peak, is more or less in the middle there, but you're seeing more variation on the acidic side. And this, again, with further analytics, has been shown to be things like deamidations that are causing your variation in the acidic. So the isoform analysis, you can see both basic variations and acidic variations. Now, moving on to electrophoretic patterns, which again is another recommendation that the ICH guidelines say you have to do and analyze and look after. We're now starting to talk about size differences. Yeah, so we've discussed charge with the isoforms, and now we're talking about size. And once again, things talk about SDS page gels and Western blots and capillary electrophoresis, but it mentions other suitable techniques as well. And what we recommend is we use capillary electrophoresis SDS, and again, this is a size-based assessment based with UV-based relative quantitation. So again, the UV relative quantitation is very good and very useful. And this is a monoclonal antibody again, and you can see 7 different batches of the same monoclonal antibody. And what you see here on the left-hand side is the non-reduced trace, and again, shows very good batch-to-batch consistency. And on the right-hand side, you're talking that we reduced the sample, similar to what Richard mentioned before, and you clearly see the light chain and the heavy chains quite nicely separated. And again, between the 7 batches, it's a very good comparability. Now, on the next slide, the next requirement from the ICH guidelines is we start talking about spectroscopic profiles. Now, in the past, up till now, with what Andrew and Richard has been saying, we've been talking about basically the primary sequence. Even when we're talking about the intact mass, the size, and the charge variation, that is still all related to the primary sequence, the actual amino acid sequence. What we're starting to talk about now is we're starting to introduce secondary and tertiary structure analytics and higher order structures. And one of the things that they mention in particular is CD and NMR. As our first go-to, we would recommend we use circular dichroism. We also would recommend to our clients that we use NMR at fairly early stages, just to try and make sure that you have an orthogonal method to function and work for you, and prove that this is the same and you've got a good quality product that you're working with. The other orthogonal method that we recommend and we use relatively routinely is FTIR, and I will give, on the next few slides, give you some examples of CD and FTIR. Excuse me. So as far as CD goes, CD can be split into far UV analysis and near UV analysis. With the far UV analysis, you're dealing with more the secondary structural analysis, and basically on the left-hand side there of the screen, you can see the trace, the spectra that you get from CD, and this is very typical for what you would expect for an antibody. That data is then taken into a secondary structural program, and you can predict the relative amounts of, say, beta sheet or alpha helix that you would get from that product. So again, with this method, you can compare your originator and your biosimilar in CD, and it gives you both the spectral output and also the structural prediction program output. On the right-hand side, we see the typical trace of a near UV analysis, and in this you're looking more at the aromatic amino acids on the outside of the protein. So you're looking more at the tertiary structure. And as you can see here, there are 2 different traces there, and it acts as a really good fingerprint to try and show that your products or your originator and biosimilar are both folded in the same way in a tertiary manner. So that's basically what you get from CD. Now, as an orthogonal method to the CD, we also perform FTIR. Now, FTIR also gives you very good secondary structural information. Now, with regard to this, what you're looking at is the 2 major peaks there, the 2 major bands there that you're seeing from about 1700 to 1550 wave numbers. And once again, with FTIR, you put this information into a structural prediction program, and you can come out with relative amounts of beta sheet, alpha helix, and this kind of thing, and then do a comparison between your originator and your biosimilar. Now, One thing to be aware of with this is that there are times when, quite often, that the algorithms are different between the FTIR and the CD, secondary structure prediction programs, and also the base data sets that they compare your spectra against are different as well. So please be aware that quite often you don't always get exactly the same numbers of beta sheet as you would expect from FTIR and CD. Now, on the next slide, the last point I'm going to talk about is the aggregation analysis, as Andrew mentioned. This is mentioned in the ICH Q6B guidelines in the product-related impurity section, and it starts to talk about dimers, trimers, and higher order multimers. Now, taking you through, again, sequentially, we've been talking about primary structure. Then I started to talk about with the CD and FTIR secondary and tertiary structure. Now we're talking about quaternary structure and how these molecules interact and align. And again, the ICH Q6B mentions size exclusion chromatography or capillary electrophoresis. And again, yes, those are fairly standard and have been used quite well. However, there are some limitations with regard to just using SEC on its own, which is size exclusion chromatography. Certainly when you're talking about larger molecules like monoclonal antibodies, once you start getting up to relative multimers of trimers or tetramers, the molecules no longer absorb the UV light that you're putting onto it. They actually scatter it. And part of the problem with that is they then disappear from your chromatogram. You can't see them. So your SEC on its own automatically removes them. So if you do have them there, you don't see them. And therefore, we recommend that you use SECMOLs. And what this is the size exclusion chromatography column system. But on the end of that, you have 3 detectors, one UV detector, an RI detector, and a moles detector, which is a multi-angle light scattering detector in series. What this does is it allows you to give and predict the idea of all the possibilities of the multimers that you may have there that you don't see with just SEC alone. Now, again, one of the other issues that was recently, probably a few years ago, found out by the FDA is that it was understood and realized that SECMOLs on its own can also have other difficulties. And one of those is that when you inject a small amount of sample down a SEC column, what you do is you have a very large dilution effect. You also can have a lot of turbidity effects and this kind of thing. What this does is if you have aggregates that are very loosely attracted to each other, you can actually pull them apart. So again, you're minimizing your aggregation. And therefore, what the FDA recommended was that you also need, as well as this column-based aggregate analysis, you needed a column-free aggregate analytics as well to be performed. And what we use is SVAUC. There are others, and I'll talk about that in the next poll question in a little while. But what SVAUC does, it's basically the opposite of what you see in a SEC column. In a SEC column, you see the dilution and the possible pulling apart of aggregates. In SVAUC, what you do is you apply a centrifugal force to the sample and push it down the cell. And again, it's UV based, so the relative amounts of, or how quickly the sample gets pushed to the bottom and how the UV absorbance changes is related to the size and the number of any multimers or monomers that you have there that are present. In this example, you see six samples of a monoclonal antibody, and as you can see, it's mainly monomer, which is good, as you would expect from a good quality product. But if you look a bit closely in the bottom side, you can see there are certain differences between dimer and trimer amounts. And so, again, it's quite interesting to be able to see which, from a orthogonal method, which you can do. Now, there is another method, and we'll talk about this a little bit in the poll question, which is field flow fractionation. Now, or asymmetric flow field flow fractionation, or AF4. Now, what AF4 does is it's kind of a halfway house between the secmoles and the AUC. Basically, you have an asymmetric flow, and the smaller molecules pass quicker along the top of the membrane, whereas the larger molecules actually, if you like, roll across the membrane a little bit more. Now, again, for orthogonal methods, very similar to the CD and FTIR, what you have to be aware of is that you will not always get the same amount of aggregation from both methods. Neither method is correct or wrong. You're doing your comparison of originator and biosimilar by SVAUC, and you're doing it also originator and biosimilar by secmoles. What you end up then is you have, if you like, as a rule of thumb, the sec or the secmoles is the best case scenario where you've got most likely to have the least aggregation, and the SVAUC is more likely to have more aggregation because of the effect that you're actually concentrating the sample as it goes down through the centrifugation process. So this brings us to our last poll question, and just out of interest, we would like to know from you, the audience, which one of these techniques below have you used to assess your aggregation of your products in the past? Have you used secmoles? Have you used SVAUC? Have you used field flow fractionation? Or have you used secmoles and SVAUC, or indeed secmoles and field flow fractionation, or in an ideal world, all of the above? So again, from me, I think it would be interesting to see what the audience, who are listening to me, whether you think you understand this and what processes you've actually used. Okay, so I think that at the end of the day, I think that's going to have to be a topic we'll get back to. So this could be very interesting. On this note, I am just going to hand back to Dr. Reason, and he will take you through the last few slides.
Thank you, Mark. Thank you, Richard, for taking us through those case studies. All I want to do now is just summarize and bring things back together and say that our services, and Biopharma Spec is a contract service provider, I hope we've shown that we operate at a high technical level. But importantly, we set ourselves up to be a collaborative partner and provide the analytical data, but also provide support from a position of experience, both with products, methodologies, etc. We keep ourselves at the forefront, and we keep the pioneering culture of the company going. Howard Morris is still heavily involved in the company, and we are consistently developing more enhanced methods chromatographically, mass spectrometrically, and with protein and carbohydrate chemistry, and always trying to push at the forefront of the analytical techniques. But importantly, for these sort of studies, we have a thorough knowledge of the guidelines and the regulatory requirements, and we have, as a team, been involved in the regulatory approval of more than 150 different biopharma products, and now approaching 60 biosimilar development and comparability programs. And I think if you are going to choose a partner, it's important to have that experience so they can really put the information into context. We do that through the analytics and through consultancy. And with that, I'd like to say thank you for listening today. The details of the speakers are on the slide, on this slide on our website. And I'd like to pass back to William now, who is going to manage a Q&A session. And on this slide are the details of the contacts in the various geographical regions if you wanted to come back to us and ask for any further information on what we've discussed today. So over to you, William.
Thanks very much for that, Andrew. Okay, audience members, it's now time for today's Q&A session. So if you do have any questions that you'd like us to ask, please send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. But for now, I'll just move on to the first question that we've received today, and that is, can I use peptide mapping to determine the structure of the glycans of my antibody?
Yeah, to an extent, the answer is yes. It will certainly give you some information about the glycans that are present on your glycopeptides. The problem that you do get with peptide mapping is that in-source fragmentation of the glycans does occur. So what that means is that the glycans will truncate. So that means that the profile that you see is not going to be a true reflection of the profile that's on the sample. So, for example, antibody glycans can truncate sufficiently to produce structures that are not actually present in the native molecule, but appear to be so because they are there in the peptide map. So whilst it is telling you something about the nature of the glycosylation, it's very important to isolate, purify, and analyze those glycans as separate entities away from the protein sequence, and use a range of analytical techniques specifically geared to the analysis of the glycans in order to characterize their structures fully.
Brilliant. Thanks very much for that response. And okay, just moving on to another question that we've just had come in. Are you aware of specific or common limitations to automatic MS assignment software packages?
Hi everybody, yeah, this is Andrew Reason. Yeah, I mean, we're very aware of that. I mean, our systems are set up to use some of the software packages for initial pass on the data. The unfortunate thing, I guess, that is the baseline for all this is that there is no software package available currently that will do a job that approaches somebody with experience in assessing and reviewing a data set. So I think if you are using some of the software packages, they can do a job. They can save you some time in terms of providing initial assessment of the data. But you have to be very, very careful in terms of how much reliance you put on that data set. And certainly that should be perused manually. That should be perused by somebody with experience. And in our services, we actually do that twice. So two people with high-end experience go through it, one as initial pass, second as a reviewer, to come up with a consensus and therefore present the data in the correct way to the correct level. Because essentially lots of these software packages do not get everything right. They get some of it right, and you have to go through it and assess it to get the true data reflection and the true assessment of the product.
Okay, brilliant. Thanks very much for that, Andrew. Okay, and it seems that we're coming to the last question of today's session, which is, when would you recommend to perform the described structural and physicochemical analytics? Mark, potentially you'd like to have a crack at this one.
Yeah, certainly. For me, I think there are several stages that's involved with that. Obviously, as Andrew said, and I think everybody in the audience agreed, that to do the initial analysis of the RMP right at the start is very important and very good. But then I think you can do it in stages when you start talking about the comparability of the structure of things. Once you've got a stable, solid process that you then know that this is going to be representative of your final drug product. Once you've got that, then you can start to do these analytics and do the comparisons, the true comparisons, hand in hand as you go along.
Brilliant. Thanks very much for that, Mark. Okay, audience members, that brings us to the end of today's session. So all that leaves me to do is to thank Andrew, Richard, and Mark for what was a fantastic presentation, and also to thank Biopharmaspec for sponsoring today's session. To the attendees, you'll receive an email shortly telling you how you can access the on-demand version of today's webinar, or you can access this through our website, which is www.business-review-webinars.com. Once today's webinar ends, today's survey will also appear in its place, and we'd appreciate it if you could just take the time out to answer the questions in there to provide some feedback. We look forward to sharing further webinars with you, so do please keep an eye out on the website just mentioned, and follow us on Twitter at BRWebinars for daily updates, and also join our LinkedIn group, Business Review Webinars. Thank you all once again, and I hope you all enjoy the rest of your